It's so easy to just look at, I don't know, a redwood tree or a bee buzzing around, or even, the person sitting next to you and think, wow, that's pretty amazing.
Yeah.
But if we zoom in, way in, like microscopically, it turns out that everything alive is built from these tiny little units called cells.
Yeah. It's mind-blowing, really, when you think about it.
And it's crazy to think about all the different types of cells.
Yeah.
Like, there are single-celled organisms, like bacteria, that just kind of do their own thing.
Right.
And then there are trillions of specialized cells in our own bodies, all working together to make us.
And each with its own specific job to do. It's a remarkable level of organization.
Absolutely. So getting a grasp on, how cells are structured and how they actually function, that's like unlocking the secrets of life itself, wouldn't you say?
It really is. And that's exactly what we're going to be doing in this deep dive today. The research you sent over is a fantastic foundation for understanding cells, these essential building blocks, and one of the most crucial processes, mitosis, how cells make more of themselves.
Exactly. So, we're going to keep things pretty straightforward today. Our mission is to basically distill the most essential information about what makes up a cell, and then this incredible process of mitosis.
Yeah.
I really want to give our listeners a clear and insightful overview. And, we'll draw directly from those sources you gave me. We don't want to get bogged down in, too much jargon or anything like that.
Yeah, for sure. We want to make this accessible and engaging for everyone.
Right. So think of this as your fast track to understanding this really fundamental part of biology. And full disclosure, you gave me some really solid, really informative stuff to work with here.
Well, I'm glad to hear that. It's definitely a great starting point for building a comprehensive picture.
Yeah.
So where do you want to begin our exploration of this microscopic universe?
I think a good place to start would be with, the basics. Let's talk about the cell itself. Just picture, a tiny self-contained world.
Okay.
Now, every world needs boundaries, right?
Right.
And that's where the cell membrane comes into play.
Yeah.
The sources describe it as this thin, flexible layer, like a selective barrier. What does that really mean? Like, what makes it so selective?
Well, I think what's fascinating here is the actual architecture of the cell membrane. It's primarily a phospholipid bilayer. So imagine like a double layer of fat molecules.
Okay.
But these molecules have like water-loving heads on the outside and then water-fearing tails on the inside.
Interesting.
And embedded within this bilayer are all sorts of proteins. And these proteins act as gatekeepers. They're channels. They're sensors. They're controlling exactly what can enter and exit the cell. Things like nutrients, essential ions and waste products. And what's really cool is that this bilayer can actually self-seal. Yeah. Which is crucial for maintaining the cell's integrity.
That makes sense.
And it's not just passive, just sitting there. The membrane is actively facilitating processes like diffusion. Think of like how perfume spreads in a room.
Right.
Or osmosis, which is how water moves across a membrane to kind of balance things out.
Okay.
And even active transport. This is where the cell actually uses energy to pump specific things in or out, even if it's against the natural flow, like pushing water uphill.
Oh, interesting. So it's working hard.
It is.
It's not just a wall.
Huh.
It's like it's actively managing the cell's needs and getting rid of waste.
Exactly. It's very dynamic.
And from the excerpt, it sounds like it's doing even more than just controlling traffic. It mentioned something about cell signaling and anchoring the cytoskeleton. Can you expand on that a bit?
Yeah, absolutely. So those proteins that we talked about that are embedded in the membrane, they also act as receptors. So they're receiving signals from other cells or even from the environment. This is how cells can communicate with each other and coordinate their activities.
Oh, wow. So they're talking to each other.
They are. And the anchoring of the cytoskeleton, which is the cell's internal scaffolding, to the membrane,
Yep.
That is super important for maintaining the cell shape, giving it structural support and even enabling the cell to move.
I see. So without that connection, what would happen?
Well, the cell could potentially lose its shape and its ability to interact effectively with its surroundings.
Right. It would just be kind of like formless.
Exactly. It needs that structure.
Okay. So we've got this dynamic outer layer that communicates, that provides structure. Now, I want to peek inside the cell. Let's talk about the cytoplasm. It's described as this jelly-like substance.
Right.
It makes me think of like cellular soup.
It is, in a way. It's mostly water, but it's also full of salts and a huge variety of organic molecules.
There you go.
And what's important to understand is that this isn't just, inert filler. It's where a ton of vital cellular processes are happening.
Like what?
Well, like glycolysis, which is the initial breakdown of glucose for energy.
Okay.
And that happens in the cytoplasm, and protein synthesis, which happens on those free-floating ribosomes.
Right.
And the cytoplasm is constantly moving, so it's facilitating the transport of materials throughout the cell, kind of like a tiny internal river system.
Like a little highway system.
Exactly.
Okay. And then right in the heart of this internal world, we have the nucleus. This is the control center.
It is.
And it even has its own, double-layered security system.
Right, the nuclear envelope.
Exactly.
It's a double membrane, and that's really important for protecting the cell's most valuable information, its DNA. And the nuclear pores, which are these tiny little channels within that envelope, they're like these highly regulated border checkpoints. They control what can enter and leave the nucleus, things like RNA and proteins. And inside the nucleus, you've got the DNA, which sometimes is in this loosely organized form called chromatin, which makes it easy to access the genetic code.
Okay.
And then sometimes it's tightly condensed into those chromosomes, especially when the cell is getting ready to divide. And the nucleus also contains the nucleolus, which is where ribosomes are assembled. So really, the nucleus is directing the whole cellular orchestra in terms of gene expression, protein production, and ultimately cell division.
So it's holding the blueprints and giving out all the commands.
Exactly. It's the boss.
Okay. I'm with you. Now, the rest of the cell seems to be like this hive of activity, and that's thanks to these things called organelles.
Right.
They're described as specialized structures, and each one has its own job.
Yeah.
It's almost like a miniature city with different departments handling specific tasks.
That's a great analogy. And the variety of organelles is just remarkable. You've got membrane-bound organelles like lysosomes, which are like the cell's recycling centers. They break down waste. And then there are non-membrane-bound organelles like ribosomes. And what's really interesting is that the types and numbers of organelles can vary a lot depending on the cell's specific role. Like a muscle cell, which needs a lot of energy, it'll be packed with mitochondria.
Okay.
Way more than, say, a fat cell, which is pretty inactive. And as you mentioned, plant and animal cells have some unique organelles that are tailored to their specific needs.
Right. Like plant cells have that big central vacuole and chloroplasts, which we'll talk about in a bit.
Absolutely.
But first, I want to kind of zoom in on some of these key players that are found in most eukaryotic cells. Let's start with the so-called powerhouses, the mitochondria. They seem pretty important.
They are essential, and their structure is really tied to their function. So they have this double membrane.
Okay.
And the outer membrane is smooth, but the inner membrane is folded into these structures called cristae.
Cristae, okay.
And this folding increases the surface area available for cellular respiration, which is how the cell generates energy.
Right.
And this process uses oxygen and glucose to make ATP, which is like the cell's main energy currency.
So it's like a little power plant.
Yeah, exactly. And here's a really interesting fact. Mitochondria actually have their own DNA.
Wait a minute. Their own DNA?
Yes. They have their own little circular DNA molecule and their own ribosomes.
So like mini instruction manuals.
Exactly. And this is actually evidence for something called the endosymbiotic theory, which is a really important idea in biology.
Oh, okay. Tell me more about that.
So the theory suggests that ancient mitochondria were actually once free-living bacteria.
Oh.
Yeah. And at some point, they were engulfed by an early eukaryotic cell. And instead of being digested, they form this mutually beneficial relationship. So the mitochondria get protection and a stable environment, and the host cell gets a reliable source of energy.
It's like a partnership.
Exactly.
Yeah.
And this little bit of DNA that mitochondria have is like a living fossil from that ancient partnership.
So it's like a reminder of where they came from.
Exactly.
Okay, so that's fascinating. So tiny power plants with their own little instruction sets.
It's pretty wild.
Okay, what about the protein factories, the ribosomes? It sounds like those are working overtime.
They are constantly busy. So ribosomes are made of ribosomal RNA and proteins.
Okay.
And they're the sites where the genetic code, which is carried by messenger RNA from the nucleus, gets translated into a specific sequence of amino acids. And these chains of amino acids then fold into functional proteins, which basically carry out every task in the cell.
So they're reading the instructions and building the proteins.
Exactly. And what's cool is that ribosomes can either float freely in the cytoplasm, building proteins that the cell needs internally,
Right.
or they can be attached to the rough endoplasmic reticulum, where they make proteins that are going to be secreted outside the cell or become part of cell membranes.
They're like independent contractors and assembly line workers all in one.
Exactly.
Okay. Well, speaking of the ER, this network of membranes sounds like another really critical part of the cell's infrastructure. We've got the rough ER and the smooth ER. So what's the main difference between them, aside from the ribosomes?
Well, the presence of ribosomes is a big clue to their different functions. So the rough ER, with its ribosomes, is all about synthesizing and modifying proteins, the ones that are destined to be secreted or become part of membranes.
Right.
But the smooth ER, it doesn't have ribosomes. It has a different set of responsibilities. So it's involved in making lipids, which are fats, including those phospholipids that make up the cell membrane, and steroid hormones. And it also plays a big role in detoxification.
Detoxification, what does that mean?
So, for example, liver cells.
Okay.
They're exposed to a lot of toxins like drugs and alcohol.
Right.
And they have a lot of smooth ER to help process and neutralize those substances.
So the smooth ER is like the liver of the cell.
That's a good way to think about it.
Okay. So rough ER is for proteins. Smooth ER is for lipids and detox.
Got it. Now, what happens after proteins are made and maybe modified in the ER, it sounds like they get shipped off to the Golgi apparatus. That's the packaging and shipping center, right?
You got it.
Yeah.
The Golgi apparatus is basically the cell's internal post office.
I love that.
So it's this stack of flattened membrane-bound sacs called cisternae.
Okay.
And it receives proteins and lipids from the ER in these little transport vesicles.
Okay.
And then it further modifies them. It might add sugar molecules to proteins, for example, to make glycoprotein.
Oh, interesting. And then the Golgi sorts these molecules and packages them into new vesicles, which are like little delivery trucks.
I see.
And then these vesicles transport their cargo to their final destinations. It could be another organelle within the cell membrane, or even outside the cell.
So the ER creates the products. And then the Golgi is like quality control and distribution.
Exactly. A well-oiled machine.
Okay, so what about storage within the cell? That's where vacuoles come in, right? Those storage sacs.
Yes. And the size and function of vacuoles can be really different depending on the cell type. So plant cells usually have this one large central vacuole that can take up a lot of space inside the cell.
Oh, wow.
And this vacuole stores water, nutrients, ions, and it also helps maintain something called turgor pressure.
Okay, what's that?
So turgor pressure is basically the internal pressure that keeps the plant cell rigid.
Oh, okay.
And it helps support the plant's overall structure. It's why plants can stand upright.
That makes sense. So what about animal cells?
Animal cells usually have smaller vacuoles, and they have more of them.
Okay.
And they're often used for temporary storage of things or for getting rid of waste products.
So big structural storage in plants and smaller, more flexible storage in animals.
Yeah, that's a good way to put it.
Okay. Now, we've talked a lot about what's inside the cell, but what about this outer layer that plants, fungi, and some bacteria have? The cell wall.
Right.
It sounds like that's pretty important for strength.
It's essential for structural integrity. So unlike the cell membrane, which is flexible, the cell wall is rigid.
Okay.
It's like armor.
Right.
And in plants, the main component of the cell wall is cellulose,
Okay.
which is a really strong and abundant carbohydrate. And this rigidity gives the cell a lot of structural support, helps it maintain its shape, and it also protects it from the high internal pressure that can build up when water enters the cell.
I see.
And that's important because that pressure is actually what allows plants to grow tall.
Yeah, that's cool. So is it just about strength, or does it do anything else?
Well, it also acts as a barrier against pathogens, like bacteria and fungi, and it also helps protect against various environmental stresses.
So it's really essential for plants to thrive.
Absolutely. It's a key evolutionary adaptation.
Okay. Now I want to talk about one last unique organelle that we find in plants and algae. This is the chloroplast.
Right.
And this is where photosynthesis happens.
Exactly. It's where sunlight is converted into energy-rich molecules.
So it's like a solar power plant.
Yeah, basically. And just like mitochondria, chloroplasts have a double membrane.
Okay.
And they also have their own DNA.
Again with the DNA.
I know, right?
Yeah.
It's another piece of evidence for that endosymbiotic theory we talked about.
Right.
So inside the chloroplast, there are these stacks of flattened membrane sacs called thylakoids, and these thylakoids are arranged in structures called grana, and the thylakoids contain chlorophyll, which is the pigment that captures light energy. And through this complex process of photosynthesis, the chloroplasts use sunlight, carbon dioxide, and water to make glucose and oxygen.
Wow.
So they're essentially fueling the plant and, indirectly, a lot of the food web on Earth.
That's incredible. So we got tiny solar-powered food factories.
Pretty much.
It's just amazing. Okay, so now that we have a good handle on the structure of the cell and these key players, I want to shift gears and talk about the cell's genetic material. You know, we often hear these terms, chromatin, chromosomes, DNA, genes.
Yeah.
Get a little confusing. How do they all fit together?
So it's helpful to think of it as a hierarchy of organization. When the cell is not dividing, which is most of the time, that's called interphase.
Okay.
The DNA inside the nucleus exists in this loosely coiled, accessible form called chromatin.
Okay.
And this less compact structure makes it easier for the DNA to be used for things like transcription, where the genetic information is read to make RNA molecules, and replication, which is where the DNA is copied before cell division. And chromatin is basically DNA wrapped around proteins called histones. You can think of it like thread wrapped loosely around spools.
I like that. So it's like easily accessible working copies of the instructions.
Exactly. But when the cell is getting ready to divide, things get a bit more organized. That's when we see chromosomes.
Right.
So to make sure that all of that genetic material gets distributed properly to the two new cells, the chromatin condenses.
Yeah.
It coils and folds up into these compact rod-like structures that we call chromosomes.
Okay.
And this organized structure makes it much easier to separate the chromosomes without them getting tangled up or damaged. And at this stage, each chromosome usually consists of two identical copies called sister chromatids, and they're joined together at a central region called the centromere.
Right, the centromere, we talked about that earlier.
Exactly. And in humans, we inherit 23 chromosomes from each parent, so we have 23 pairs.
46 total.
Yep. And these chromosomes carry all of our genes.
Okay, so chromosomes are like the neatly packaged, transportable version of chromatin ready for distribution.
Exactly.
Okay. Now, what about DNA itself? That's the fundamental instruction manual, right?
Absolutely. DNA, deoxyribonucleic acid, it's that famous double helix.
Yeah.
It carries all the genetic information in specific sequence of its 4 nucleotide bases: adenine, thymine, cytosine, and guanine. And DNA is mainly found inside the nucleus. It directs the development and functioning of an organism, basically by providing the instructions for building proteins. And of course, it's the molecule that gets passed down from parents to offspring.
Right. So it ensures the continuity of life.
It does.
Okay. And within that really long DNA molecule, there are specific segments, the genes. These are like the individual units of heredity.
Right. So a gene is a specific segment of DNA that carries the code for a functional product. And that product is usually a protein, but sometimes it's a functional RNA molecule. And these gene products determine our traits and carry out all the functions necessary for life. Genes can vary in size, and their activity, whether they're on or off, is very tightly regulated.
I see.
It depends on the cell's needs, its stage of development, and the environment. And the human genome is thought to have somewhere between 20,000 and 25,000 genes.
Wow. Okay, well, that gives us a much clearer picture of how the genetic material is organized. So now I really want to dig into the main event, mitosis. This is how cells divide and create 2 genetically identical daughter cells. So why is this process so important?
Mitosis is absolutely essential for life.
Okay.
In multicellular organisms like us, it's the basis for growth. It's how we develop from a single fertilized egg into a complex organism with trillions of cells. It's also crucial for tissue repair. So when you get injured, mitosis replaces those damaged or lost cells.
Okay.
And in some organisms, especially single-celled eukaryotes, mitosis is actually how they reproduce. And the key outcome of mitosis is that each new daughter cell receives a complete set of chromosomes that's identical to the parent cell. So it ensures genetic continuity.
So making exact replicas.
Exactly. And this intricate process happens in 4 main stages: prophase, metaphase, anaphase, and telophase.
Right.
Do you want to break down what happens in each stage?
Yeah, let's do it.
Okay, so let's start with prophase.
Let's go.
This is the first stage, and it's where the cell starts to visibly prepare for division. Remember that diffuse chromatin we talked about?
Yeah.
Well, in prophase, it condenses into those distinct visible chromosomes. Each chromosome consists of those 2 identical sister chromatids joined at the centromere. At the same time, the nuclear envelope, the membrane that surrounds the nucleus, starts to break down. This allows the chromosomes to move freely. And a structure called the mitotic spindle, which is made of microtubules, starts to form.
Okay.
And two centrosomes, which help organize the spindle fibers, move to opposite poles of the cell.
So it's like the cell is setting the stage for the division. Now, we talked about the centromere before. What's its role in all this?
The centromere is really important. It's that constricted region on the chromosome where the two sister chromatids are held together. But more importantly, it's where the spindle fibers attach.
Oh, okay.
So there's a protein complex called the kinetochore that assembles at the centromere.
Okay.
And the spindle fibers physically attach to the kinetochore. And this connection is what allows the spindle fibers to pull the sister chromatids apart later on.
I see. So each daughter cell gets a complete set of chromosomes.
Exactly.
Okay. And what about the spindle fibers themselves? What are they doing during this whole process?
So spindle fibers are made of microtubules, which are part of the cell's internal skeleton, the cytoskeleton. And these fibers extend from the centrosomes, which are at opposite poles of the cell. They do two things. Some spindle fibers attach to the kinetochores on the chromosomes, and others interact with spindle fibers from the opposite pole. The ones that attach to the chromosomes are responsible for moving and separating the sister chromatids, and the fibers that don't attach to chromosomes, they help the cell elongate. Basically, they prepare the cell for dividing into two daughter cells.
So they're the ones that are actually doing the work of moving everything around.
They are. They're like the movers of the cell.
Okay. So once everything is set up with the spindle fibers attached, we move on to metaphase.
Right. And metaphase is all about alignment.
Okay.
The spindle fibers are pulling on the chromosomes from opposite poles, and this tension causes all the chromosomes to line up in the middle of the cell.
Oh, wow.
Yeah, it's pretty amazing. And this line is called the metaphase plate. And this alignment is super important because it makes sure that when the sister chromatids separate, each new cell gets a complete set of chromosomes. It's like a final check to make sure everything's in the right place.
So it's like you're all lined up for inspection.
Exactly.
Okay. So then the actual separation happens in anaphase.
That's right. Anaphase is action time. It starts with the sister chromatids suddenly splitting apart at the centromere.
Okay.
And once they're separated, each sister chromatid is now considered its own chromosome.
I see.
And these new chromosomes are pulled to opposite poles of the cell by the shortening spindle fibers. And at the same time, the cell itself elongates even more, getting ready to be split in two.
So the identical copies are pulled apart and the cell is stretched out.
Exactly.
Okay. And then in telophase, the final stage, we just need to put those chromosomes into new nuclei.
Right. So in telophase, the chromosomes arrive at their poles and start to uncoil. They go back to that relaxed chromatin state.
Okay.
And then a new nuclear envelope forms around each cluster of chromosomes, creating two new nuclei.
Okay.
And each nucleus has a full set of identical genetic information. And then the spindle fibers break down, and that completes nuclear division.
Okay. So we've got two nuclei, each with a complete set of DNA. But we still have one cell.
Right. So that's where cytokinesis comes in.
Cytokinesis.
This is the actual physical division of the cytoplasm. It usually starts in late anaphase or early telophase, so it overlaps with mitosis a bit. And it's a little bit different in animal and plant cells.
Okay.
In animal cells, the cell membrane pinches inward, creating this cleavage furrow.
Okay.
And that furrow gets deeper and deeper until the cytoplasm is completely divided. In plant cells, it's a bit different.
How so?
Because they have that rigid cell wall, a cell plate forms in the middle and grows outward, eventually becoming a new cell wall that separates the two daughter cells.
So pinching in animals, building a wall in plants.
Exactly. And the end result is two new genetically identical daughter cells.
Now, we've been talking about sister chromatids and daughter cells. Can we just clarify what those terms actually mean?
Absolutely. So sister chromatids are the two identical copies of a chromosome that are made during DNA replication.
Okay.
They're attached at the centromere until anaphase, and they each contain the same genetic information. And daughter cells are the two new cells that are produced by mitosis and cytokinesis. They each get a complete set of chromosomes and organelles that are identical to the original cell.
Got it. So mitosis is a really important part of a bigger process, the cell cycle. Can you tell us more about that?
Sure. So the cell cycle is basically the entire lifespan of a cell, from when it's formed to when it divides into two daughter cells.
Okay.
It's a continuous process, and it's really tightly regulated. And there are two main phases, interphase and the mitotic M phase.
Okay.
Interphase is much longer than the M phase, and it's when the cell is doing its normal activities.
Okay.
It grows, it duplicates its DNA and organelles, getting ready for division. And the M phase is when mitosis and cytokinesis happen.
Okay.
And the cell cycle is controlled by checkpoints, which make sure that everything is happening correctly.
So it's like a really carefully choreographed dance.
It is. And those checkpoints are super important for preventing errors that could lead to things like cancer.
Right. So they're like the quality control team. Okay. We talked about diploid cells earlier, the ones that have two sets of chromosomes. What about haploid cells? Where do they fit into all of this?
So diploid cells have two complete sets of chromosomes, one from each parent. Most of the cells in our body are diploid. In humans, the diploid number is 46. And haploid cells have only one set of chromosomes. In humans, our gametes, which are the sperm and egg cells, are haploid.
Okay.
And when a sperm and egg fuse during fertilization, they create a diploid zygote.
Right. So that's how the diploid number is restored.
Exactly. And this fusion of genetic material from two parents is really important for genetic diversity.
Okay. So diploid for our body cells, haploid for reproductive cells.
Yeah.
That makes sense. So to sum it all up, we've taken a pretty detailed look at the structure of the cell, the functions of its organelles, and this amazing process of mitosis, which is how cells create copies of themselves.
Yes, we've covered a lot of ground.
It's really incredible to think about all this happening on such a tiny scale.
I know, and it's happening in every living thing.
Yeah, it's mind-blowing. So for our listeners, I want to leave you with this thought. We've seen how intricate and carefully controlled this process of cell division is.
Right.
So what happens when things go wrong? Even small errors in this process can have a huge impact on an organism's health and development. It really makes you appreciate the delicate balance of life.
Absolutely. And what we've talked about today is just the beginning. There's so much more to explore about cells and how they work.
Oh, yeah, for sure. And we'll definitely be diving into some of those topics in future deep dives.
I'm looking forward to it.
Me too. Well, thanks for joining us today as we took this deep dive into the world of cells and mitosis. Keep those questions coming and stay curious.
Thanks for having me.